Well, hello, everyone. Welcome to the CCMB seminar. It's my great pleasure to introduce our speaker today, Dr. Tsang from NIH. John is a senior investigator in the Intramural program of NIAID at NIH and chief for multiscale systems biology section. He's also co-director for the NIH Center for Human Immunology. John is a computer scientist by training. He received his bachelor's degree in computer engineering at the University of Waterloo in Canada and received his PhD in biophysics from Harvard. After graduation in 2008, he went to Merck in Boston as a senior scientist for 2 years before joining NIH. John has pioneered for the computational systems biology studies of human immune variability. And his work has been recognized by multiple awards, including the NIAID Merit Awards in 2014 for his outstanding contribution to the influenza vaccination studies, and in 2017 for the leadership in developing a platform for large-scale data reuse, and 2020 for his scientific leadership on COVID-19 study. I think he's going to share with us some of this work today, and I'm really looking forward to hearing about it. So without further ado, I think I'll pass over to John, and welcome, John.
Thank you so much, Joan, for the introduction, and thanks again for the invitation. So just to avoid bandwidth issues in the middle, I'm going to stop my video for now, but feel free to ask any questions along the way and stop me, okay? So today I'm going to tell you mostly our work in systems human immunology. Mainly looking at both the human population level as well as single cells. So let's see. Yeah, so just before I get into the specifics, I want to sort of define a bit on what is systems biology and immunology, because still, even at this day and age, people think of it, see it as like a different level, sort of like, or they have different definitions. So to me, there's sort of two types of systems biology. There's the top-down kind, with the premise that there's much that we don't know yet. So the goal of this kind of an investigation is to discover the components and their interactions and start to build black box, for example, machine learning, predictive models of the outcomes and systems behavior. The other type, it's what I call bottom-up emergent phenomena, systems biology, with the premise that there's much that we can intuit, right? So I'm sure many of you appreciate how complex biology can be when you stare at a diagram of, for example, molecular and cellular interactions. It very quickly would escape our intuition of what might happen if you think about things over time and over space, for example. So that requires more quantitative models of how these different components actually work together to give rise to these emergent phenotypes that can be very complex, as I would describe it, for example, in humans. So in this talk, I'll mainly talk about the former. I actually try to fit in some of the bottom up stuff as well that we do, but it turns out that the talk will be really quite too long. And I also want to cover some of the COVID-19 work that we've been recently been doing that Cho mentioned. So therefore, I will quickly highlight a little bit on the bottom up aspect, but most of the talk will be focusing on the former. So systems biology really built on sort of two pieces. One is involves technologies for measuring better and measuring more, so that we can really take a look, comprehensive look at cellular and molecular states of biological systems, for example, before and after a perturbation. The other, it's really the development and application of computational and mathematical approaches for both the processing, integration, and modeling of these data, so that we can learn some new biology. And hopefully we can also get to some useful models that are hopefully predictive as well. So the immune system is really a nice system to do systems biology and to think about systems biology. And when you think about it as a system, right, it has a wealth of inputs. For example, the amount of antigen, giving, for example, a lot of you may have received the COVID-19 vaccines. So it the signal, the information that's carried by the mRNA molecule instructing the system to make proteins. So that's, you can think of that as an input. And how that presents over space and time, and how the environmental factors, like for example, whether there's something called adjuvants, which is something that would drive inflammation in your system. So those are all kind of inputs to your system. And then you got outputs, things that can do work, right? So you got, for example, antibodies that emerges, and also T cells, CD8 T cells, for example, that actually kills cells that got infected by the virus. And then you also got memory. So these are encoded primarily by adaptive lymphocytes that can remember the encounter, such that in the future, if you encounter something, the same pathogens again, those cells would get expanded again and can combat the infection. And in the middle, there's this processing, right? And that's where you have these very complex interactions among molecules and cells and over tissue environments that would give rise to these outputs. So just a little bit more detail, when you think about at a high level how vaccines work, it sort of involves like sort of three major steps involving different types of cells. In the beginning, there's like the detection of these vaccine components, and that would also activate the innate immune system. So there are cells that encode pattern recognition receptors that senses some of the molecules in these vaccines, and they can get activated. These cells will also take up these molecules, these antigens. And so, for example, by professional antigen presenting cells like dendritic cells, they then move to secondary lymphoid organs like lymph nodes to start activating other cells. So, for example, they would interact with T cells, and by these dendritic cells can present antigens to the T cells and educate them. And these activated T cells can then also help activate B cells, and these B cells can turn into these fairly large antibody production and secretion factories, basically. And then some of these cells, T and B cells, would differentiate and turn into long-term memory cells. And for example, they reside in your bone marrow, and they can really stay there for years and years. And that's how successful vaccines work. It's really by generating that kind of memory. And then there's the response, both the more acute kind, the antibody response that gets generated that can help protect you. And if the pathogen comes in, you're coming in contact with the pathogen, and these antigen-specific B cells. Again, memory is a key outcome of vaccination. So my lab sort of works in three areas of systems immunology. First is really human immunology. So a key question we ask is, why do individuals in the human population mount variable immune responses? So we also conduct fairly a comparative analysis of human immune states across health and disease, so to better understand common pathophysiology across different diseases, and also to define metrics of immune health. So for example, here at the NIAID, we have taken advantage of a large number of rare disease patients. So these are almost, you can think of them as human knockouts. So we have these monogenic disease patients with basically genetic defects at various key genes that function in the immune system. So we have conducted a comparative analysis of these patients across more than 25 such diseases, and also looking at healthies. And that allows us to start to define what it means to be healthy by looking at so many diseases. So the sort of second area we work on, it's computational and also experimental approaches to enable systems immunology. So for example, we develop single cell analysis approaches. We also develop new techniques for building predictive models. We have also been developing and maintaining a crowdsourcing platform, I think that Tsang mentioned in the beginning, to enable data reuse, integration, and analysis, particularly for non-computational immunologists. And the third area we work on, it's the more bottom-up kind, and specifically in single cell biology, and with a focus on understanding the biological function of cell-to-cell variations. So as mentioned, I won't go into the details in this talk, but I'll highlight a few examples here. So in case you're interested, you're welcome to look at some of the papers, or please send me an email. So one question is, we're very interested in understanding the function of single cell variations. So even in very homogeneous populations, So in this work, highlighting, for example, we link human population variation, so basically person-to-person differences, to single-cell variations in these people in specific T-cell subsets. And we found that the single-cell variations within a person can be associated with human aging, and these variations, independent of the mean expression levels of a protein or a gene, they can also be shaped by the genetic variations known to cause disease in the human genome. As a second example, here we use human macrophage as a model to study how the environment shapes gene expression variation across single cells and how that variation can propagate in signaling in gene networks. And motivated by the observation of the macrophage work, we also developed a framework called MEPA that basically trains machine learning models to more accurately predict phenotypes emerging from gene network dynamics. So when you look at like cells, basically the underlying molecular networks can generate all sorts of gene expression variations as well as like correlated variations over time. So we try to understand how that kind of phenomena sort of come about, but that typically requires fairly expensive stochastic computational simulations. So here we basically bypass this kind of expensive simulation to allow us to explore at a very large scale the parameter space and asking what kind of parameter combination would give rise to specific phenotypes in these single cell variations and also correlations. And more recently, in collaboration with Harry Wong from Ron Germain's lab, we use this approach MEPA to study the homeostatic control of single autoreactive T cells. So it turns out that in addition to having autoreactive T cells pruned during T cell development in the thymus, you can actually find plenty of T cells in circulation that's actually reactive to your own tissues even. But there's also quite a bit of peripheral control, like basically there's circuits that limits the number of these cells from being activated. So here we actually built a multi-scale temporal spatial model of interactions among the dendritic cells, the conventional CD4 T cells, and regulatory T cells in lymphoid organs, and study how this kind of feedback circuit can constrain the number and the activation level of these self-autoreactive T cells. So it's a very nice piece of both quantitative modeling and machine learning and experimental, a lot of imaging work. So this work is actually coming out and very soon in Cell. So and you can take a look at the details. So now I want to turn your attention to human immune response variations. So as I mentioned, immune responses, it can be really highly variable across individuals. As you can see from the current COVID pandemic, for example, right? So if you look at, for example, cancer, people receiving immunotherapy, so only about 30% of metastatic melanoma patients, they actually respond to anti-PD-1. And if you look at vaccination outcomes, antibody responses to influenza vaccination that we typically get each year, they can span hundreds of folds across people, and they also can be different between different age groups, for example. And if you look at autoimmunity, let's take an example like lupus, right? So if you look at different lupus patients, they go through these periods of clinical quiescence. So they look like very healthy, normal people, but they can also flare up with disease activity, so with inflammatory activities. And these can be highly variable from patient to patient. So the question we're really interested in to answer is include whether such immune response outcomes can be predicted, and if so, what those predictive parameters are, and can we start to point out what are the determinants and start dissecting potential mechanisms, and also whether there are shared predictors and mechanisms across different types of immune responses. Did I hear a question? Yes.
Sorry, yeah. Can I interrupt here for a question?
Yeah, of course.
So just curious, the young population seems to be more variable than the older population. Is there any reason?
Yeah, so in general, the young, because they typically respond quite well. So quantitatively, you can see that they have a huge dynamic range of responses. And most of them mount a response that's reasonably protective in the sense that they have an antibody response level that crosses a certain threshold. for so-called neutralization. While in the O, especially in the elderly, their responses are typically quite poor, actually. So they're much more compressed towards the lower end, and actually most of them don't respond. And then there are a few individuals who respond well. And that's why, like in the case of flu, for example, the elderly individuals get a much higher dose of the vaccine. And folks are also experimenting with like adjuvants to try to boost the responses. So in a way, the O is basically mostly non-responders, while the young are mostly, they respond, but the quantitative, the quantitative range of their responses are highly variable. And that's actually something that we are trying to understand, is why is it that some people can mount such a very gigantic response while others are much lower. Yeah.
I think that's.
So that was a great question to get to this next slide. So a lot of factors can contribute, obviously. So including genetics, and there's intrinsic factors such as like sex. So I'll get to that in a little bit, but females, they tend to mount stronger responses, for example. And also pre-existing immunity. Some individuals have like higher levels of pre-existing memory cells, for example. So that can shift the response depending on what kind of vaccines and pathogens or perturbations. But based on twin studies, so folks have looked at like twins, right? So it's quite clear that genetics alone is really far from explaining such variations. So for example, if you look at flu again, these are in adults. Antibody response to flu vaccine have zero heritability in adults. So that means that if you look at a pair of identical twins, you vaccinate both, and one can be a really high responder, like the one in the 500 range in terms of fold change in antibodies. The other one could be much lower, for example. So the effects of pre-existing immunity is also quite complex in humans. So if you measure, for example, the level of B and T memory cells specific to influenza at the baseline before you get a vaccine, they typically tend not to correlate well with the antibody responses at all. So there's something else going on. So another important contributor we found a number of years ago, it's the status of the immune system before the perturbation. So back then we did the study where we enrolled these healthy individuals. We took their blood 7 days before they got the vaccine, and also right before they got the vaccine. And so the blood allowed us to extract various immune cells from these individuals. And then we gave them the vaccine and then monitored their day 1, day 7, and day 70 time points following vaccination. So the day 1 allow us to look at their so-called innate, like very early responses. The day 7 allow us to look at their sort of adaptive, like B and T cells. And the day 70 is a longer term time points to see whether the system reverted back to baseline or not. And from these samples, we generated a lot of data. There's like transcriptomic data generated from these cells. We have the antibody response data from these individuals. We have single cell phenotyping using flow cytometry, which allow us to look at the various, like what type of immune cells they are, like memory cells, activated cells, effector cells, and intermediate developing cells and whatnot, right? We also have data on do these B cells respond to certain specific flu antigens. So the question remains, it's going back to Chong's question about these responses are very variable in terms of antibodies. Why is that the case, right? So we found things like age and a little bit of sex effect, but one of the surprising things that we found at the time was that the status of the immune system before they got the vaccine really matters. So we found a set of cells that were measured in an antigen non-specific manner, basically, that if they have higher levels of certain cells in circulation, they respond more highly to the vaccine. And the other concept that we uncover in this study is that those parameters, they tend to be very individualistic in that if you look at their day -7, so a week before, or even the day 70, they tend to be quite stable within these individuals. So suggesting that it's a fairly, at least within the timescale of months, these are temporally stable, right? And so they're maintained over time. So I'm also giving you a hypothetical example of what happens if these are not stable, So you can see that these individuals are intermixing now. They're mixing with each other. So therefore, they cannot really characterize that particular person. So, so the next question is this general, right? We found this in kind of one cohort. So to see whether that's the case, I'm going to skip some details. We computationally derive a surrogate, basically a set of genes that are really tracking whose expression in the blood is tracking the frequency of these cells over in this cohort. We then tested it in independent human cohorts, multiple influenza vaccination studies from different locations and years. We also wanted to see whether this is something more general than looking at flu influenza responses. So we looked at another study where they have a different vaccine, and that's a yellow fever vaccination. So it's actually a live virus that these people got. But the good thing about this live virus is it's attenuated, so it actually generates a response that's protective, but it doesn't actually hurt you. But it's a totally different set of antigens and a different kind of vaccine, basically, that these guys got. So first, the results just blindly just apply that baseline signature into these influenza cohorts. These were the results we got. As you can see, that we see a consistent trend where the high responders based on our predictor, it's always responded with higher antibody levels in these different cohorts. And when we look at yellow fever, we saw the same thing, basically, that this particular baseline state also delineated higher responses to yellow fever, which, as I mentioned, is a totally different and independent vaccine compared to the flu, basically. So our data basically suggests the existence of this baseline immune state that can be shared across vaccines and even in the live viral infection, basically. And the yellow fever result further suggests that our baseline signature does not merely reflect some sort of prior exposure or antigen-specific immunity, because the vaccinees were all naive to the yellow fever to begin with, because yellow fever in this country, you typically never see people got infected by them, basically. So then the next question is whether the same signature may actually be predictive in the context of disease flares, not in autoimmunity, right? So that's where we kind of trying to broaden this out further to think about whether this is relevant in even a totally different context, basically. So for many autoimmune diseases such as lupus, as I mentioned, one of the key conundrums is that the predictors and determinants of these flares, they remain fairly elusive. So especially those that can predict disease activity using immune parameters during clinical quiescence. So if you look at a person, when they're pretty healthy, like, can you say something about how badly their disease activity will be in the future? So this is actually distinct from uncovering signatures of disease severity or disease activity. So it's quite well known, if you look at lupus patients, for example, that many of them have a so-called type 1 interferon signature in the blood. So these are, and so that's well known in these patients. But it's unknown whether its status during clinical quiescence can actually be predictive of flare intensity. So to assess the signature I described to you for flu vaccines in this context of flares, so we use a longitudinal blood transcriptomic study from lupus patients. So in this cohort, the patients, based on the original study, actually, they can be grouped into several distinct molecular subtypes based on their blood transcriptomic status during the flares. So one particular group of patients, in this case, I'm highlighting groups 2, 3, and 4 here. So basically these groups. So each column here is a patient group, and each row corresponds to a transcriptomic phenotype. So interferons, for example, myeloid cells. So I want to highlight this particular signature. It caught my attention in that they display a very strong plasmablast or B cell signature during their flares. So the reason why this is interesting is because this actually resembles how a healthy person responds to a vaccine. So when you give somebody a vaccine, typically 7 days after they got the vaccine, you can see a surge of these cells in the blood, these plasmablast cells, basically. So this set of patients in lupus, they display the same kind of signature, right? So therefore, a compelling hypothesis was that if you look at these patients at their clinical quiescent time points, that's sort of like a baseline, basically. Would this be the same as in a vaccination situation where the state at these clinical quiescent time points can allow us to start to predict or understand why some of these patients would flare highly, basically, right? So that was the question. So, so shown here is a single patient over time. So, and the y-axis is the disease activity of these patients. So by disease activity, I mean, in these patients, you can find symptoms like skin rashes, or even in the severe ones like kidney inflammatory situations, they have kidney problems, for example. So these patients would have these kinds of they would have high flares and coming back down again and going up again, right? So to test our hypothesis, what we did is we computed our sort of TG6 score at these at the low disease activity time points, like the blue ones here, and then for the high ones and the red ones, we computed the plasmablast score, basically. So then we asked whether the baseline state in these patients is predictive or correlated with these increases in those patients. So this was indeed the case for the specific patient groups with the plasmablast signatures I've shown you. So on the left are those patients from patient groups 2, 3, and 4, or even more specifically in patients 2 and 3, because those were the patients with the strongest plasmablast signatures. And on the right, there are the rest of the patients, and we didn't see any correlation, basically. So this is quite exciting in the sense that lupus is a very heterogeneous disease. So in a way here, we're pinpointing in a specific subset of patients with a well-defined molecular phenotype during their flares, we're starting to better understand one, why and what kind of predictors we may have for why they're flaring up highly. And second, it's now we can start to drill down on what that signature may mean, and that may point to some potential therapeutic and also clinical monitoring options. So this is the next question then, right? So what does this signature, mysterious signature, really reflect? So to look at this, this is now a few years ago now, we use CySeq. It's a single cell analysis approach to start to profile these high and low responders. So we started out with the vaccine responders first to try to see with this kind of whole blood transcriptomic signature at the single cell level, what do they really reflect, basically? Because in blood, you get all sorts of immune cells, so it's hard to tease out exactly where the signature came from. So CySeq, it's now a few years old, and so what it provides, it's cell surface protein expression and also transcriptomic measurement at the same time in single cells. And recently, we can also measure VDJ sequences, so allow us to trace TCR and BCR, for example, in lymphocytes. And like flow cytometry, CySeq uses antibodies to measure the protein expression, typically on the cell surface. But instead of using fluorophores in flow cytometry, so oligo barcodes are conjugated to these antibodies. And by sequencing, we can read out many of these proteins. So this is one of the earlier panels we designed, measuring 82 proteins. But in recent work, we have gone up to about close to 200 to allow us to measure really a variety of cell lineages and also states in the population of cells. So we also work out these approaches to really multiplex that a lot to allow us to look at multiple individuals over space and time, and also to save money. So for example, if you got individuals coming in and you have multiple samples collected from these individuals, you can pull them together where individuals are identified by SNPs. So we actually use the sequencing data to call SNPs in these individuals, and by pulling them together, we can use the SNPs to demultiplex the individuals back out. And then the time component, it's barcoded by antibody hashing barcodes, basically. So this allows us sort of a two-dimensional sort of multiplexing. So we can pool individuals, we can pool time points, and now we can really generate data from close to 40 samples all together in one shot. We also developed some techniques because this kind of data generates some unique challenges, especially at the lower level. In terms of protein noise, they're quite distinct from typical single-cell RNA-seq kind of data. So here we conducted both experiments, but also quite a lot of computational analyses to tease out sort of two major sources of noise in these kinds of protein expression data. And so we also have an open source package you can use called DSB, R package, that if you generate this kind of data, you may find it very useful. So and so typically in flow cytometry, we sort of use manual gating, right, to define the different kinds of cells. But with so many proteins, we can do fairly well sort of unbiased analysis to just cluster all of the cells, especially at the protein level. So one thing I want to highlight by using the proteins in particular is that for immunologists especially, there's a lot of knowledge built over the years on what these different surface protein markers mean in terms of what kind of cells they may mark. So it's rather advantageous actually to really utilize these proteins as much as possible to define these cells. So we did it here, for example, like we clustered the cells at different resolutions to sort of review different cell subsets. So you can see the major immune cells at this level, but then they can be further clustered into subsets and finer to review these states, activation states, and so on in these cell populations. And on the right, I'm just showing you a map of the sort of like with these different populations and clusters of cells, what the protein phenotypes look like, right? So you can start to see like some of these B cell markers and some specific DC and stem cell markers and marking different subsets. So using this data, what we are now able to ask is, if you go back to that signature, we remember I call that TG6 signature, those genes that mark that baseline state, we can now zoom in and ask, in which specific clusters of cells can we see that those genes are different between the high and the low responders, right? So one thing that caught our attention very early on is we see that these cells, these genes, they tend to be expressed at a higher level in high responders in a population called PDCs. So these are so-called plasmacytoid dendritic cells. So so that's very interesting because these cells, they can make they are a primary factory of these alarming molecules called type 1 interferons, basically. So usually these cells are only activated after you get an infection or after some certain tissue damage, for example. And the fact that in these very healthy young individuals, you can see that there's a contrast between some individuals having more elevated PDCs in terms of these genes versus others. So that was very intriguing. So we hypothesized that maybe this reflects the fact that in some people, these PDCs are already more activated. So therefore, we tested, we derived some signatures of dendritic cell activation independently from some other data and also existing knowledge, and tested whether these cells are indeed more activated. And we found that, yes, that's indeed the case, that these cells, if you look at the high responders versus low responders at the baseline, they are significantly different, basically. So, and then we look into the subsequent lymphocytes and other cells also. So I don't have time to really show you all the data, but basically further deep dive into the single cell data suggested a sort of a two-stage immune response model. So even at the baseline, the high responders, they tend to have a temporally stable, more activated baseline immune state. So, so we saw more activated PDCs, as I mentioned. So these guys basically, even at baseline without any perturbation, likely led to these higher levels of type 1 interferons. And therefore, we detected broad elevations of so-called type 1 interferon signatures across both myeloid and lymphoid cells, right? And we also saw that, again, without any overactivation, these T and B cells in these people are already persistently more activated at the baseline. And these are unlikely specific to particular antigens because of their frequency. So they're likely quite polyclonal, basically. So what's intriguing is that despite this kind of poised state, you actually need a trigger. So a trigger is actually needed to get to the second stage of the response. So in this case, like I've told you an example, and you need a vaccine or you need some flare trigger in the case of autoimmunity to drive this kind of full-blown antigen-specific response, resulting in these B cells and antibody production, right? So what we actually don't know still, it's what sets such a temporally stable baseline set point state, right? What exactly is maintaining this temporal state in these individuals? So one potential source we look into is these chronic viral infections that some of us have, for example, like CMV. So that's something that about 50% of the population have these kinds of latent dormant virus in the system. So interestingly, these folks with CMV, they are marked by an elevated levels of these T cells called TEMRA cells. So we look into that and we didn't see any differences in these high and low responders. What about sex, right? Because I mentioned earlier that females, they tend to mount higher responses to some vaccines. And that actually, we saw something interesting. So indeed, you see that if you look at this baseline state score, the females, they tend to be higher than the males. But if you look into the females only, you still see that the low, middle, and high responders are still segregated. By the low, middle, and high, I mean their response to a flu vaccine, basically. They're still segregated by this score. And same thing in the males, right? So suggesting that, again, this baseline state that we uncover involving these PDCs and these poised activated T and B cells, they can delineate they can still basically predict the response independent of sex. So it's a fairly general baseline predictor that goes beyond the sex of the individual. And another intriguing thing is there's another paper that came out. There might be some endogenous priming signal in the system. So for example, this recent paper in mice, they show that the microbiome can actually maintain a more elevated PDC state, basically. So perhaps something in our gut, it's persistently sort of setting our system at a certain level. So that's one potential intriguing answer. So just want to conceptually summarize. So first, so far, I think I've shown you that the baseline set point predictors and potential determinants, they can be shared across vaccines and infections with the flu vaccine and the yellow fever infections, right? And second, our data also point to a sort of maybe a personalized approach to monitor and treat these lupus patients. So for example, there are PDC-based therapies in development. So perhaps by dampening these PDCs at the clinically quiescent states, we can avoid flares in these patients. So our finding also suggests a more of a shared set point mechanism for both vaccination, infection, and autoimmunity. So now I want to turn your attention to sort of COVID. So the work I'm going to tell you really next took really a village. The experimental data and analyses I'll present here were primarily led by Ken Liu, Andrew Martins, William Lau, and Nick Rachmaninoff in my lab, with help from many others, including Jingguo Chen from the Center for Human Immunology. And the COVID patient samples, they came from Brescia, Italy, during the early pandemic. So here's an overview of the cohort, human cohort I'm going to focus on today, mainly showing their hospital stays. So the x-axis is time. Each row is a patient, and so as the time it's measured since the symptom of onset. So when they experience disease symptoms. So for most of these patients, we have at least like 2 time points from their hospital stays. And the severity classification here is mainly based on measurements of oxygen levels, basically, and also the organ functions. So here, the patients we have, they bias towards the more sort of critical scale, because one, they were hospitalized, and second, in the early pandemic situation in northern Italy, when the hospitals were really overwhelmed, only the most severe patients got admitted to the hospital. So given the large number of patients classified as critical, the first thing we did is so here you can sort of see each row here is a patient, and each column correspond to various lab and also circulating protein parameters, right? So you can see that even these critical patients here, they have very heterogeneous molecular sort of phenotypes. So therefore, the first goal we had was trying to see whether we can establish a more quantitative disease severity metric, a score, using these kinds of parameters. So we did that, and so the thing we saw was that by using machine learning to sort of fish out which one of these parameters may be more predictive of the severity categories based on just clinical information, right? We found that these ones I'm highlighting here in the red box, they turn out to be the most informative. I'm not going to go into the details on what they reflect, but suffice to say that by using these parameters, we can construct a fairly continuum sort of metric that can define the severity of these patients. So we then validated that this metric is actually meaningful and relevant by using an independent cohort of 67 patients. So here you can see that the disease severity metric is significantly different across the severity scales, or this is the so-called disease ordinal score. You can see the extensive heterogeneity in this critical patient group, right? So, and furthermore, this metric is also associated with ICU admission status. So it can actually predict when a patient came in, who's going to go into the intensive care unit ward. Again, in this totally independent patient cohort. So, and this is true even when you look at only the critical patients. So these patients all came in with a very high requirement of oxygen. But you can see that by using this metric, we can now say that certain patients are more likely to be in the ICU because they are more severe, according to this molecular quantitative definition, basically. So in the following analyses, we basically use this DSM metric exclusively, so that we have a continuum to look at these patients. We also bin the patients using their DSM scores into a balanced high and low groups using the median value. So I already introduced CySeq. Basically, in this case, we did this almost like on steroids in a way. We basically took immune cells from these patients and profiled them by CySeq, but using an updated panel with 192 markers. And in addition to protein and mRNA expression information, we also sequenced their VDJ, so allowing us to look at the TCR and BCR in the lymphocytes. And the other experimental detail is that aside from just looking at the circulating immune cells, we also sorted the non-naive B and T cells, which allow us to enrich for the clonally expanded cells. So this allows us to get data from more than 400,000 single cells after QC, and using the proteins again, we can start to look at the zoom in and look at the transcriptome of 29 higher level cell types and subsets. We also identify more than 100 higher resolution cell subsets, but I won't go into the details of those. So our sort of question focused on two key dimensions of the immune response in COVID-19. One, it's what defines severity? Like what are the immune response sort of signatures associated with disease severity? The other is how the response evolve over time. So this time component is very important, and how that really differs between patients with different severities and outcomes. And that could really help us, I think, we hope, to identify critical points of clinical monitoring and intervention. So I apologize for this very busy plot, but the high level sort of message I want to convey is that we can identify these very cell type specific. So the columns are the different cell populations and immune cell types, and the rows correspond to various cell states and processes. And again, this is really focusing on the earlier time point when these patients first came into the hospital. So that we basically fitted a model that accounts for age and sex and the time since symptom onset to derive this map. So you can see a side-to-side comparison on first the signatures that's distinguishing patients versus healthy controls. And on the right-hand side, it's the same kind of map, but it's looking at association with disease severity, right? So you see that while type 1 interferon antiviral signatures are elevated in these patients, as expected, they actually inversely correlated with disease severity in these patients. And so they're more depressed in the most severe patients. So which is kind of counterintuitive, but I'll get to that a little bit. And as you saw earlier, there's many associations. Like for example, if you look at this map on the right, there's many bubbles that pass statistical significance. But if you try to construct sort of a map on what are the variables that may be connected to disease severity, you end up with kind of a hairball kind of graph, right? If you look at each node as a transcriptional signature within a specific cell type, and if you draw the edge as a correlation measure, you get this kind of like map basically on the left. But this is not super informative typically, right? Because it's just too many things and everything is correlating with everything else and could be driven by lots of hidden variables. So without going into the methodological details, we derive an approach that basically assesses more in statistical independent relationships. So we try to do sort of conditional independence statistical modeling. So to arrive at this kind of like simpler picture where we try to find really what are the key or primary direct associations of disease severity, basically. So here we actually got decent strong evidence for actually just 4 of them, basically. So again, these are cell type specific states that we found that are connected to disease severity. So one that we found was this, again, it's involving these PDCs again, it's very interesting, which is a cell death signature in these cells. So that partially explains why some of these patients have this kind of lower type 1 interferon or antiviral signature, because actually in these more severe patients, these cells that are making those interferons, they may be dying actually. So there's a strong death signature. The other 3 are all negatively associated with disease severity. And they're inflammatory signatures or fatty acid metabolic signatures in these NK cells, basically. So I'm going to elaborate on those a little bit more. And so what we saw was quite surprisingly that both this metabolic signature, these fatty acid biosynthetic and oxidative catabolic genes, they're elevated in the most severe patients. So this subset of NK cells is known to be rapidly activated by a cytokine called IL-15 in early viral infections. And in these patients, IL-15 is actually persistently elevated in COVID patients. So therefore, we look at IL-15 in circulation and found that it's actually strongly associated with disease severity in these patients. And also, IL-15 is also significantly correlated with these strange metabolic signatures that we found. So suggesting that perhaps this very persistent exposure to IL-15 may really be driving this phenotype. The other thing we saw was that the inflammatory genes, for example, those downstream of NF-kappa B, they tend to be negatively associated with this fatty acid gene signature, as well as interferon gamma. So these are typically markers of inflammatory activation in these cells. So overall, these data suggest that in these most critically ill patients, these NK cells, they might actually be in this kind of more burnt out, low inflammatory state, despite that the external environment is actually very highly inflammatory with these IL-15. So basically, they've been exposed to the environment for long enough that they are getting into this kind of like, you can call them exhausted or tired kind of state, basically. So finally, one thing I want to highlight, it's the time component. So if you look at the trajectories, we found a number of parameters that seem to peak or start to go into different directions around this day 17 to 23 after symptom onset period. So here's an example involving some cells that are peaking basically around day 20, right? So suggesting that this might be an important period in the immune response. We found the same thing if you look at like neutrophils, for example. So then we specifically tested what happens, like are there any dynamical shifts around this juncture period? So we found that the inflammation in cells like the NK cells I highlighted and also in the monocytes, they actually flip. So they went from being lower in the most critical patients, as I showed you earlier, to now going up higher, basically, in these patients. And that's intriguing. And then we also look at the circulating proteins. So these are inflammatory mediators. And you sort of see a similar picture in that in red are the more severe patients. You can see that by the time they hit this kind of period, they start to inch up a little bit in some patients, but in the milder cases, they actually start to go down a bit, right? So to really further test this concept on whether around this time there's something crucial happening, we hypothesized that the immune status of the patients at this period might actually inform outcomes. Outcomes as in, for example, if you look at these critical patients, some of them will proceed onto recovery, and some actually, unfortunately, would die. So therefore, we actually go back to the larger cohort and get circulating protein cytokine data from two sort of large age and gender matched subcohorts with either recovery or fatal outcomes, basically. And so what we did is we tried to ask whether we can see what are the proteins or markers that are shifting basically right around that period. And we found a very statistically significant number of such proteins, particularly involving these pro-inflammatory molecules like IL-6. So for example, in IL-6, what happens is that by the time you hit the juncture, these critical patients are, when you compare those who proceed onto fatality versus those who recover, they start to exhibit large differences in the level of IL-6 at this time period compared to before, right? And then there's also other ones like IL-17 or even markers of maybe perhaps inflammation and other types of things that are basically divergent in around this time point. And you can see the same thing also even after you look at the post-juncture period versus before. So there's basically there's a temporal shift. And in fact, if you look at the markers at this time point, you can actually predict fairly well on who's going to proceed onto death versus recovery. So this is sort of the AUC curve of that prediction by using these markers. So yeah, so if you look at their sort of trajectory reconstructed by piecing together longitudinal time courses within these patients, but also borrowing information across patients, you basically see why that procedure I described before uncover things like IL-6 and IP-10. So you can see that some of these patients, like these disease patients, they start to have elevated IL-6 in these fatal patients, while in the ones who recover, they kind of like maintain or some of them even go down depending on which proteins you look at, right? So this point is sort of like represents a point of divergence in a way for these patients with different outcomes. This actually paper just came out a couple of weeks ago, so you can look at the details in that paper. But just time for a really big picture summary. So we see that early on in the disease course, around maybe day 7 to 15 after symptom onset, we see this unexpected and attenuated inflammatory signature, such as lower type 1 interferons and this IL-15 link kind of exhausted burnout phenotypes in these NK cells. And these generally correlate negatively with disease severity, right? So one question, as I mentioned before, is what might be driving this attenuation? So one hypothesis based on the analysis was that some of these cells making the relevant inflammatory molecules like type 1 interferons are actually dying. The other one is there's also this sort of negative feed-forward loop that's going on. So patients who experience strong inflammatory activation earlier on, they typically would make negative feed-forward molecules to dampen feed-forward response, so that naturally evolution basically would try to make sure that we don't mount an overt response, because typically inflammation itself can kill us, right, basically. So that actually could be a reason. It's actually a natural good response to suppress the system to make sure it doesn't go into overdrive, right? So that's been shown actually in some mouse models in using flu infections as a model, right? So a key question, it's really what drives this potential second wave that I have shown you. So a number of factors can be involved, and they could be quite different from patient to patient, basically. So for example, there could be increased tissue damage. There's more danger signals that drive that inflammation. There could also be, in some small number of patients, some secondary infections and even bacterial infections. So finally, another question is whether this attenuated, potentially more burnout state earlier in the time course might actually be a problem. So they are insufficient to handle some of the perturbations that these patients experience in the second wave, and therefore they had this kind of poor outcomes. So with that, I just want to thank again folks in my lab. I think I highlighted the individuals along the way and on who did the work, and then really the vaccine work and the baseline set point work I highlighted earlier was really led by Yuri and Rachel Sparks in the lab, and along with the Center for Human Immunology. And I already mentioned the COVID work. And finally, I just want to mention that we're sort of like, I told you quite a lot of work based on blood, but we're really diving into tissues and try to understand immunity at the tissue level, and how responses at the tissue level may also reflect into signatures in the blood. So if any one of you are interested in thinking about along these lines, we got support from the ZZI on doing these, and also there's other sort of ongoing projects and sort of future directions that we're heading into. If you're interested, please feel free to reach out. So we're building some in vitro models, and really in humans, as you may imagine, it's not easy to access tissues. So therefore, having in vitro models of immune responses is important to understand what we just can't observe in blood. And we're also starting to engineer cells. And the great thing about immune cells is they can go to lots of places. So they can both act as sensors and also as effectors. So to do that, we are starting to develop approaches to really start to predict and engineer some of these molecular recognition and also immune cell interaction kinds of engineering that we're doing. And also all these single cell data are snapshots, but we also have real time in some of these projects I mentioned. So we're also inferring quite a lot some of these data, like immune cell dynamics. We also are setting up a sort of independent fellows program, providing independent resources and funding, and also with initial personnel. So again, if you or any of your fellows are closer to finishing their PhD or adventurous in heading into some of these directions, please reach out. Okay, I think I've gone a bit over time, but thank you so much for your attention, and happy to take any questions.
Yeah, thank you so much, John. This is very interesting. Let's open the floor for a couple of questions. If anyone has any question, please feel free to unmute your mic.
I see something in the chat.
Yeah, if people can also check the. So I can start with a question, John. I was curious that for your COVID work, have you looked at any patients that are being recovered and then have second infection? Do they have different immune response compared to their first?
You mean like recover from COVID and then they got COVID again?
Yeah, I think there are quite some cases out there.
Yeah, in Brazil, especially, it's very infamous now. Yeah, they have these variants that seem to be reinfecting individuals who got COVID from another variant before.
So you think it's
from the same variant, I'm not aware of a lot of cases like that. Yeah.
Okay.
So, but a related answer to that, it's if you look at vaccination responses of patients who got COVID before, after the first dose of a Pfizer vaccine, as you would expect, some of these recovered patients can mount very, very strong responses to the vaccine. And there's some anecdotal also descriptions of their adverse reaction. Some of them can mount quite a bit of maybe more inflammatory responses to that first dose, for example. But these I don't think have been looked at, like, broadly speaking, like yet. I think there are some real data coming out on how these prior COVID positive patients respond to the vaccines. Yeah.
And what about the different regions? Have you guys getting data from different areas, maybe for different variants?
Yeah, so we have not looked at the host immune response to the different variants yet. Some colleagues are looking at that. I suspect that the basic trajectory dynamics are likely quite similar, except I think the cases that you described where you have COVID positive to one strain and then now you have another variant coming in, and how does the response differ? I think that's very intriguing. I don't think we have a lot of data on, by we I mean the broadly speaking, have a lot of data on that yet, basically. But I think some of those may emerge pretty soon on these, like if you're immune to one variant and then now you're infected by another variant, what happens kind of situation, basically. And the most worrying are ones from South Africa and the Brazil so far, because if you look at their the antibodies from those, let's say you look at the antibodies respond to either the vaccine or the previous wave of COVID, the neutralization capacity of those antibodies are quite a lot lower to these new variants, basically. So, yeah.
I saw a question from the chat. It's quite interesting. So have you looked at the childhood immune signatures? When do you think the immune signature set point you identified is established during the development?
Yeah, that's a great question. So we have some data looking at that. It's a very interesting, it's a very complex issue. So we see that, like, when you look at infancy, the immune system develops very rapidly. So the cell composition and all of that changes quite rapidly. I didn't have time to show you some data, but we have generated some of these single cell CySeq and also some ATAC-seq data on very early on in the first year of life, and also then over time. But then if they slow down, and then we can start to see both age-dependent shifts, as well as things that got set very early on, and they remain very stable despite development in these people, basically. And we're still in the process of teasing out whether the set point that I described, it's something that's being set, like the question was alluding to, like basically almost like within the first year. And then they're basically set for that individual. Or at later times they can be shifted, and we found perhaps maybe a mixed answer so far, and in that it looks like it could be set fairly early on, but certain exposure and other events could actually change things basically later on as well. So that's why I'm not quite sure about the answer, and we may need to expand the population a bit to really at least the population locally here to understand better what's going on. And I also want to mention that geographic differences are very significant as well. So if you look at like an endemic population to a certain pathogen in, let's say Africa, they have a very different types of baseline immune set points than what we found in North America. And the nature of it can be different, and the implication of it can also be quite different.
Do we have any more questions?
Hi, John. This is Tankarpola. Very nice work and lots of data. I want to take a little bit of a different perspective and ask you, you focused on blood for very good reasons, and yet you're interested in going into tissue. And I guess going into it, we don't really know what information we're missing. In other words, we know what we see in blood. You established a pre-existing state, which sounds promising in having some sort of causality in driving different future responses. Does the molecular nature of that pre-existing state lend itself to any understanding of whether it's self-propagating, in other words, something where the blood itself is maintaining that state, or whether it's a response to something coming from tissue, some other source? Is this something that you can resolve?
Yeah, that's a very good question. We are looking at that a bit in mouse models and try to see whether, of course, it may not reflect what's going on in humans. So here's are some speculations in terms of humans. It's that I think it's most likely originating from tissues. In that, if you look at the circulating PDCs in blood, they're super low in frequency. The only reason why we were able to detect a signature in the past, when we look at like bulk blood, like if you just extract RNA from blood, it's because they have these downstream effects on the T and B cells. So these more poised, more activated lymphocytes. And they also respond to the elevated type 1 interferons, and therefore you can see a type 1 interferon signatures as well. So that's what allows us to detect a signature. But as you trace it back in using the single cells, for example, that we look at, luckily we can start to pinpoint the potential origin of that, which is likely the PDCs. And the PDCs are much more abundant in tissues and in secondary lymphoid organs. So most likely these PDCs are either shaped by the local tissue environment, or they're remembering something. And so the mouse example involving the microbiome, it's one sort of example where the microbes in the gut, it's setting the status of those PDCs. And in a way, they're kind of setting the individual's sort of interferon set point in a way, in that particular example. So that also lends itself to this kind of a theory on that it's likely originating from the tissues. And finally, we have some preliminary data showing that a prior infection can shift the sort of the both structurally, but even the architecture of the tissue in a way that there may be some residual states that these cells may now be in a loop that keeps secreting certain molecules. And that can actually impact future responses, at least through the same route, for example. So whether that kind of, you can call it training immunity or however you want to call it, it does leave a fairly permanent scar, like over a time scale of months in a mouse. So a month in a mouse is like many years in a human being. So that's another example. Yeah, but for that particular human situation with this potential PDC set point, we don't have a definitive answer as to one, the origin, and two, whether it's causal, in the sense that if you really go in and now perturb it. We have some evidence that some vaccines can actually leave a person in that kind of more elevated state for quite long periods. So it's suggesting that it could be causal. But again, we need to really do these over long-term experiments to figure that out. That was a long answer to your, but very good question. So,
is there any other question from anyone? If not, yeah, please join me to thank John for this wonderful talk and great. Yes, thanks everyone for coming to the seminar.